Very good morning to all of you. My name is Dr. Santosh Upadhyay, and I am an assistant professor at the Department of Biotechnology, Kuma University campus, Bhimtal. And in this lecture, I'm going to talk about the basics of hybridoma technology and production of monoclonal antibodies. Okay, so let's start. So first of all, the question is, what is the difference between monoclonal antibodies and polyclonal antibodies? Okay, so if you can see, this is an antigen, okay, and an antigen is made up of different epitopes. An epitope is the part of antigen to which a particular antibody binds. Okay, so an antigen is normally made up of multiple epitopes. Okay, so when you inject this antigen to mice, okay, or any other animal, maybe human, okay, antibodies are raised, okay, against different epitopes, right? So numerous B cells make different kinds of antibodies, okay, and these antibodies combinedly known as polyclonal antibodies. Why? Because number one, they are made up from different B cells or different clones of B cells, okay, right? And the other thing is normally they binds with different epitopes also, okay. So this is called polyclonal antibodies, okay. But think of if one of the cells, if any of the B cells producing these antibodies, suppose that this B cell, the central one, okay, this becomes transformed. Transformed means getting cancerous, right? So what will happen? This will divide and redivide to make identical cells, and this population of identical cells would be known as clonal population, okay. And this clonal population, because they are derived from a single cell, they would be producing identical antibodies. They would be called as monoclonal antibodies, okay. And let us talk about what is the procedure of monoclonal antibody production, or what is a hybridoma, okay. So in the previous case we just talked about one cell getting cancerous, or one B cell getting transformed, okay. But that may not happen as per your will, right? So what is usually carried out, because these clonal population of B cells can be useful for getting antibodies or monoclonal antibodies of uh, desired specificity, right? So we need to produce such cancerous B cell, okay? Those are producing our desired antibodies. So for that, a method was devised long back, okay, around 1975, I think, by two scientists we'll talk about, okay. And in this one, what you normally do, a mouse is challenged with the antigen, okay, and now the spleen cells are harvested, okay. So we get a population of B cells, okay. And then what we do, we take myeloma cells. Myeloma is a kind of cancer of B cells, okay. We take these cells because they are cancerous cells. They have got they are kind of immortal in a way, right? Okay, if you provide the nutrients, they keep growing, right? So then what we do, we fuse the B cells or the spleen cells with the myeloma cells. Both of them are basically originally B cells, because myeloma cells is a cancer of B cells, but they have got certain properties. Number one, they are cancerous. Number two, they are not antibody producing anymore. They are not producing their own antibody anymore, okay, though they are derived from B cells, okay. Now when we fuse them, what happens, in this population there would be myeloma cells, there would be spleen cells, and there would be fused cells, okay. So that fuse, because when the certain cells would be fused with the certain of the myeloma cell would be fused with the spleen cells. So these fused cells would be known as hybridomas. Okay, and you know what happens? What is the great property of these fused cells of hybridomas? These fused cells actually produce the desired antibody. Okay, and that property is coming from B cells, and they are immortal as well because the myeloma cell part of them make these hybridomas immortal. Okay, because they are a fusion of myeloma cell and B cells. Okay, but there is a problem because this is a mixture. If we talk about this while we carry out fusion, this mixture contains three types of cells: spleen cells, myeloma cells, and fused cells also, or hybridomas. So how to remove these B cells and these myeloma cell from from the hybridomas? We want just hybridomas now. Okay, so there is a medium called het medium. That is a very important medium. You would need to study about it. Okay, we'll talk. We'll give a some idea of the het medium. The het stands for is named after three of its components: hypoxanthine, aminopterin, and thymidine. In this het medium, only hybridomas do survive. Myeloma cells, B cells are killed. They die because they don't have property of indefinite growth. Myeloma cells are also killed somehow, and we'll talk about the mechanism how they are killed. Okay, so ultimately only hybridomas survive. Now we grow these hybridomas in bulk inside a culture vessel or inside a inside an animal. Okay, we can inject these cells in an animal, and they form tumors, and then we can from their serum we can harvest these things, CITES basically, and then these are the monoclonal antibodies. The hybridoma was discovered by Kohler and Milstein, okay, or we can say invented by in 1975, and they got Nobel Prize for medicine in 1984 for this development of hybridoma. Okay, so what are the basic requirements for hybridoma production? Number one, a well purified antigen. Okay, because we would be needing antibodies against some antigen, right? We will take that antigen and immunize an animal. Okay, and then B cells are isolated from the spleen of immunized animal. Okay, now we take myeloma cells. So myeloma cells we already talked about. Okay, they are B cells, kind of B cell lines, but they have lost antibody producing capability, right? And besides, they are also they have also mutation in few of their genes, like the HGPRT or TK. There are two genes. Any of them should be mutated. Why it is so? We'll talk about. Okay, and then we would need some fusogen. What is a fusogen? They are the agents those promote fusion of B cell and myeloma. Okay, and the fusogens could be chemical fusogens like PEG, polyethylene glycol, and biological fusogens like Sendai virus. Okay, so Sendai virus also kind of promotes fusion of B cell and myeloma. Okay, and then when the fusion happens, then what we want? We want specifically the hybridoma. Okay, from a mixture of B cell, myeloma, and hybridoma, we just want hybridoma. For that, head selection is used. Okay, so now let us talk about what is this het medium and what it does. Het medium contains three components: hypoxanthine, aminopterin, and thymidine. Okay, all these cells, when we grow them in culture, they need to make different components, different they need to make proteins, they need to make different fatty acids, need to make nucleotides, etc. Okay, so what happens for B cells? All of their biosynthetic pathways are normal. In myeloma cells, there we said that okay, the myeloma cell is mutated in TK gene or HGPRT gene. What does this HGPRT gene do? HGPRT gene is required for making nucleotides in a salvage pathway. Actually, for nucleotide biosynthesis, there are two pathways. One is called de novo pathway, where your cells start from scratch and make the required nucleotide. Okay, so de novo pathway, that is called de novo pathway. And then there is a salvage pathway, where you provide them with certain precursor molecules. Okay, and by using these precursor molecules, they convert them to nucleotides by using certain genes like HGPRT or TK. Okay, so what is hypoxanthine? That is used as a nucleotide precursor in salvage pathway, and HGPRT gene is required for its utilization. Okay, so our myeloma cell is HGPRT negative. That means if our myeloma cell is HGPRT negative, means it cannot use the salvage pathway, or it cannot use the hypoxanthine for synthesis of nucleotide. So if we are growing our myeloma cell in a het medium, so in a het medium, say the myeloma cell is not having the HGPRD gene, so it cannot use the salvage pathway of nucleotide biosynthesis. Okay, now it depends solely on the de novo pathway, but the other component of the het medium is aminopterin. So what is aminopterin? That is a inhibitor of de novo pathway. So for a myeloma cell, if we are growing the myeloma cell in a het medium, this de novo pathway is already blocked because aminopterin is a poison for de novo pathway. It's an inhibitor of de novo pathway. Okay, so it should be dependent on precursor molecules to synthesize their nucleotides by using salvage pathway. But the precursor molecule is there. Of course, in the het medium, the precursor molecule is there. Hypoxanthine is there. Though there should not be a problem, but the problem lies inside the myeloma cell because they are HGPRT negative. Okay, they are HGPRT negative. Their HGPRT gene is mutated. Okay, either HGPRT gene or TK gene, thymidine kinase gene. What is third component? That is thymidine. This is also a nucleotide precursor for the salvage pathway. And for that, for its utilization, TK gene is required. Thymidine kinase gene is required. So our myeloma cell is either mutated in TK gene or in HGPRT gene. Okay. So myeloma cell by itself can never grow in a het medium for long. It can never grow for long in a het medium because the aminopterin blocks its de novo pathway of nucleotide biosynthesis, so it cannot make its nucleotides. Okay, through the de novo pathway. Of course, hypoxanthine and thymidine are there. Hypoxanthine and thymidine are there for their utilization by salvage pathway. But you know what? We have deliberately selected the myeloma cells as TK negative or HGPRT negative. They are mutated in HGPRT and TK, so they cannot use hypoxanthine and thymidine either. Okay, so the in a het medium, myeloma cells cannot survive because their de novo pathway doesn't work, their salvage pathway cannot work because of these mutations. B cells never a problem because they have a limited lifespan, so they just die after some time. Okay, what about hybridoma? These are hybridomas, those only survive in a het medium. Why? Because, of course, their de novo pathway is blocked. Okay, de novo pathway is blocked both for B cells and myeloma cells in a het medium. Okay, right? So now let us talk about the salvage pathway. You know, in this because this is a fusion, hybridoma is a fusion. So the HGPRT and TK gene is mutated for the myeloma cell partner of this fusion, right? But it has also got the nucleus of B cell. Nucleus of B cells means the genes of B cells, right? Okay, and in the B cell, the HGPRT and TK both are functional. Okay, so the hybridoma can actually utilize hypoxanthine and thymidine for synthesis of nucleotides by salvage pathway. Hybridoma cannot utilize de novo pathway because aminopterin is there, but it can utilize hypoxanthine and thymidine for by using salvage pathway for the synthesis of nucleotides because functional copies of TK and HGPRT genes are complemented by the B cell. Okay, so hybridoma has got antibody producing activity. It has got a capability of indefinite growth also. Okay, and we already selected them by using the het medium. So this het medium, the mechanism of het medium is very important, and you should be learning that. Okay. Now, what are the application? Diagnostic application. Nowadays, you must be listening about different ELISA methods or even other methods used for diagnosis of COVID-19. Okay, ELISA is used for many diseases. It's a very simple technique. Enzyme linked immunosorbent assay you must be knowing about. Then radioimmunoassay. Okay, this is very sensitive assay, but it uses radioactivity for detection. Then immunohistochemistry. In a histological slides you can see the presence of different molecules, maybe viruses, maybe pathogens, okay, or different proteins. By using these monoclonal antibodies, those could be labeled with certain enzymes, and then you can develop color reactions. Then therapeutic applications, because in cancers there are different growth factors. If you block those growth factors or their interaction with their receptors, we can possibly inhibit the growth of cancer cells. Okay, so you must have heard about herceptin. Okay, that was a monoclonal antibody device against the HER2 receptor in breast cancer. Okay, there are actually a whole list of such therapeutic monoclonal antibodies now. Those are being used in different cancers. Okay, then against different inflammatory diseases like your arthritis and other diseases where you need to control the inflammatory molecules like TNF alpha. So antibodies against TNF alpha or other pro-inflammatory molecules is used for neutralizing those pro-inflammatory molecules. Similarly, angiogenesis related diseases like cancer and some ocular diseases, okay, where the a lot of angiogenesis or blood vessel formation takes place in your retina. Okay, so by using the antibodies against angiogenesis promoting molecules like VEG, a vascular endothelial growth factor, you can actually control these things like cancer and this retinal angiogenesis. In science, because we need to explore different molecules, their positioning, their location, their interaction. Okay, so for the analytical application, we also use monoclonal antibodies like for Western blotting. You all are aware of Western blotting. Then IP and co-IP, that is immunoprecipitation. Then you can from a lysate of a cell you can do IP, and then you can carry out co-IP, co-immunoprecipitation, to examine the protein-protein interaction, right? And then there is another technique called CHIP, that is chromatin immunoprecipitation, that is used to analyze the interaction between your protein and different DNA molecules. Okay, so in this also we use antibodies against different protein molecules. Okay, and then those protein DNA complexes are immunoprecipitated, and then those specific DNA molecules are identified using different methods like PCR. Okay, and there are preparative applications also where we use monoclonal antibodies for affinity purification of different molecules. If there is a complex mixture and we want to just isolate one molecule from there, we can use monoclonal antibodies, okay, and we can pull that down. So this is all about the hybridoma, monoclonal antibodies and their applications. We would be covering the detailed aspects of hybridoma technology and applications of monoclonal antibodies second lecture. So thank you so much for your attention.